So we have already studied that there are two approaches that can be used to locate the gene within the genome of a particular organism. And we have studied one of the approaches, that is the statistical approach. The second approach is based on the similarity of the two genes. And this approach is based on the fact that any of the protein that is sequenced might be related to the protein that is already sequenced of any of the organism. But the drawback of this approach is that we cannot use that approach because we have already seen that the structure of the genes, especially if there is a protein, we can do it, but if we are dealing with the genes, that we have seen that the structure of the genes, that is intron, exons and other structural organization of the genes, they are different in different organisms. So if the gene is sequenced and we cannot use that newly sequenced gene to compare with the already sequenced genes of the other organisms. And the example is that 99% of the human genes are almost similar to the genes of the mouse that is present in the genome. So we can simply look for a similar sequence based on the genes known in the another organism. So in the previous strategy, in the statistical analysis, we search the specific regions that are present at the junction of the exons and introns. And we know that these structures that are present at the junction were not conserved, and we may lead to the wrong information of the introns. So the common things that produce is that within the organism that the genes and in turn their proteins that are being produced in two different organisms, they are the same. But the structure, organization of the genes, they are different in different organisms. So in this similarity approach, we can use the computational puzzle. In this puzzle we will use or in this approach we will use the substrings of the gene, and then we will analyze the substrings with the substrings of the other organisms. For example, if there is a gene in the human, we will first divide the gene into the exons, and we consider exon as a substring of the gene, and that substring will be compared with the substring of the same gene in the other organisms. So instead of employing a statistic analysis of exon, similarity-based method attempt to solve the combinatorial puzzle, find a set of substrings, as we have already told you, in a genomic sequences whose concatenation fits a known human protein. In this scenario, we suppose we know a human protein and we want to discover the exon structure of the related gene in the mouse genome. The more sequenced data that we will collect, the more accurate and reliable similarity-based methods become. So consequently, the trend in gene prediction has recently shifted from statistical motivated approaches to similarity-based algorithm, because these similarity-based algorithms, they are more reliable as compared to statistical analysis approaches.